Hi, everybody. Thank you to the organizers for inviting me, and thank you to all the participants for this opportunity to share some of my work with you. I'm going to be talking today about an efficient way to do multi-objective optimization for neuronal cell and circuit models. And so the real question here is, well, how can complex models be efficiently optimized to match experimental constraints? And so in this talk, I'll go over some approaches to model optimization that have been commonly used in neuroscience, some introduction to the use of parallel computing for model optimization. And then I'm going to introduce a new parallel computing tool that's called nested, which can be used for multi-objective optimization of neuronal cell and circuit models. And then I'll show you an application of this tool to a biophysical and morphological single cell model, and then an application to a large scale biophysical and morphological network model. So here's the situation that we find ourselves in as modelers frequently, that a new experiment has been done and has collected some really interesting data that tells us something about the biology. And we want to incorporate that level of detail into our models so that our models can make more accurate predictions. And so here's one of my favorite kinds of experiments, because it's near and dear to my heart. I care about how neuronal dendrites process information. And so here's an experiment that was done in Heinz Beck's lab, where a recording was performed at the cell body of a neuron, and another recording was performed out in the dendrite of the cell. And so when a current injection into the soma evokes an action potential, you can measure the amplitude of that spike in the dendrite. So these spikes backpropagate into the dendrites. And if you do this many times in different neurons, recording from dendrites at different distances, you can see that there's some fall off in spike height with distance from the soma. And so we could build a morphological model of this cell. We can reconstruct the morphology of the cell, and then we can perform simulations of recordings at different dendritic sites. And we can evoke an action potential at the soma and look at how the spike height attenuates. And so the types of parameters that we might want to vary to try to get this model to be accurate could be the density of sodium channels in the dendrites. These would amplify spikes. And in fact, if there was a very high sodium channel conductance out in dendrites, we might have no attenuation at all. We might be able to propagate a full height spike. And then there'll be potassium channels in the dendrite, and these will act as a brake on excitability, and they could potentially reduce the propagation of spikes into the dendrite. And so what approaches should we use to do this kind of optimization? So, I mean, typically, especially when there's a low number of parameters and it's a system that we have a lot of domain knowledge about, then we can sort of use, be domain experts, and maybe we've already have measurements from immunohistochemistry or from single channel recordings that there are potassium channels in dendrites and sodium channels in dendrites. So we can kind of just start with what we know and see if it works. And so you can see in this case, the experimental data is in these black points, and this dotted line is a model simulation where the dendrites are passive. So if there's no sodium channels and no potassium channels in the dendrites, actually, we're already kind of in the ballpark to reproduce this data set. But we could also vary these parameters. So if we add a little bit of sodium conductance into the dendrite, you can see that you're still in the ballpark. Maybe you're overshooting a little bit. The cell's a little bit excitable, but maybe we could add a potassium channel and we could counterbalance this. So really what we need to do is kind of have a bit more of an unbiased screen of what the possibilities are here. And so we could take this, two parameters, we could divide it up into equally sized bins within some experimentally defined bounds. And so we could test, okay, here's a range of sodium channel conductances in dendrites. Here's a range of potassium channel conductances. And then we can measure the attenuation of the spike at some location in the dendrite, say at 150 microns away from the soma. And what we find is that there's actually a lot of combinations of parameters that pretty accurately recapitulate the experimental data. So this is model degeneracy. And this could also be telling us something about the biology. Maybe there's a lot of heterogeneity. Different neurons would come up with different solutions to the same problem. But so we can learn something about the possibilities from doing this model screen, this parameter screen. But in this case, we had just two parameters. We divided up the parameter space into 20 values, and so we only had to simulate 400 iterations of this model to try to get an answer about what this parameter space looked like. But if we increase the complexity of our model, so here's another hippocampal dentate granule cell model, where now there are more species of ion channels that are considered. There's multiple types of potassium channels, there are calcium channels. And then it was also considered that these channels are expressed at different densities in different compartments. So here, sodium channel density is very high in the axon, so that spikes are actually initiated in the axon initial segment. And then there are gradients of these channels into the dendrite. So the proximal dendrite has a different density than the distal dendrite. And so, conservatively, we've got maybe 40 free parameters now here. And even if we did a very coarse grid search where we divided all of these parameters into 10 possible values, then what we're looking at is, even if we could get our simulation time to, a millisecond, it would take us 10 to the 30 years to complete this grid search. So we obviously need a more efficient way to screen through parameters for these complex models. And so there are approaches that involve computing a gradient, so trying to look at the shape of the error landscape of your model, and then non-gradient based approaches. So if you're really lucky, the sensitivity of your objective error to your parameters is nice and smooth. And so you can sample a couple of points in your parameter space, and you can directly compute a gradient or a slope, and then you can follow that slope down to some global minimum. And then you've got sort of the optimal best answer for how your model can be fit to experimental data. But it's often the case really with these detailed biological models that the features that we're trying to measure are maybe not even continuous themselves. They jump in they make discrete jumps. And there are correlations and nonlinearities and sensitivities to the parameters that just make for a very bumpy error landscape. And if we were to just sort of blindly use a gradient-based approach here, we would start somewhere, and maybe wherever we start, we would end up with a different answer. But we would follow the slope of this error function, and maybe we would find ourselves stuck in some local minimum, but never be able to find a global solution. And so there are a couple of approaches that have been used in neuroscience to try to do this kind of parameter search without relying on an explicit gradient. And one of them is called basin hopping, or simulated annealing. And what we do here is we sample a set of parameters, we evaluate a cost function or an objective error, and then we have a temperature term, which essentially determines the step size. So initially we could start off with a very high temperature, which means we can jump around and make very large hops within our parameter space. And if we happen to find a model that performs better than the previously sampled models, then we can make our hops from that location, and we can reduce the temperature over iterations until finally we're making smaller and smaller hops, and we're kind of polishing up our solution. And so like the initial phase of making large hops at high temperature should try to find all of the basins, and then the final steps of polishing should be able to sort of drill you down into what's hopefully a global minimum. And this can work quite well. Another popular approach is a population-based optimization that is a common evolutionary and genetic algorithms take advantage of this type of approach. And what you do here is you generate a whole population of models that have different parameter configurations. But then rather than computing sort of the absolute cost function or the sum of all of the objectives that you care about, you can actually compare the models to each other in the population. So what you're looking for now is relative performance improvement compared to the neighbors in the population. And so you can have a selection criterion that actually is comparing to the other members of a population. And then you can actually consider the parameters themselves to be like nucleotides in a genetic code. And you could take two models that performed quite well, and you could perform crossover where you say, okay, you take my sodium channel conductance, I'll take your potassium channel conductance. Let's see whether we can produce an offspring that performs better at our objectives. Or you could take one of the parameters and you could perturb the value, and that's kind of like a genetic mutation. And so this is an iterative algorithm that would keep generating populations and evaluating them relative to each other. And then hopefully in the end, you'll end up with a good performing model that sort of matches your objective criterion. But models have many features, and we have to run many tests to be able to evaluate all those features. And how do we optimize multiple objectives? So here is an example from the Allen modeling group of a biophysical model of a layer 5 pyramidal neuron. And there was experimental data where current was injected into the soma with different amplitudes. Different protocols were used, like a ramping stimulus or a noisy stimulus. And so we want the model to be able to reproduce all of the responses to these different kinds of stimuli and probes. And an evolutionary approach was taken. So this was a population-based approach. And so at every generation, a bunch of models would be generated and evaluated relative to each other. And then over iterations, performance improved on all of these features. And then at some point, actually additional tests were added to the test set, and then performance improved even further. And so what you end up with is a model that in response to these kind of somatic current injection stimuli resembles the experimental data quite well. And this kind of approach typically requires a lot of model evaluation. So each generation here contained around 1,000 individuals, and it proceeded for 1,000 generations. So in the end, we evaluated on the order of 1 million models. But there's another problem, which is that when you're in this multi-objective regime, some of the features that you're measuring might actually conflict with one another. And so we want there to be a global solution. We want to find the best tuned model, but in these multi-objective cases, really what you end up with instead is a family of models that are all kind of performing to different degrees on different features. And so here's like kind of a diabolical case where every time that improvement was found on this performance criterion on the x-axis, it led to a worsening of the performance in another dimension or another feature. And if we took the average of these two cost functions, or if we summed up our objective errors, we would have just a whole family of models that are really indistinguishable from each other. But this one's the best performer on one objective, and this one is the best performer on another objective. And maybe here's a model that balances them pretty well, but it's not the best at either one. And so there's an algorithm that you can use to try to identify all of the models that live along this surface, which is called the Pareto front. And it involves this pairwise kind of comparing your performance to your neighbor. And so if you take one of these models and you ask, is there anybody else in the population that is equal or better than me at all of the objectives? And there are other models that are performing better or equal on all of the objectives. And so that's called domination. So if you're an individual model that is not dominated by any other member of the population, then you probably live close to this Pareto front. And we want to try to highly rank all of the models that are not dominated. But another thing, so then let me just go over some sort of approaches to parallel computing that can be used when you have lots of models that you want to evaluate. So if we had n models to test and we use a serial approach, this would be like gradient descent. You could basically sample one model at a time and compute your slope and follow your slope. Or basin hopping was originally proposed as a serial iterative algorithm. So it takes one processor and you test one model at a time, and then you'll complete in n steps. If you were performing a grid search, or if you could sort of pre-compute, these are all of the models that I want to test, I don't need it to be iterative, then you kind of know ahead of time, and you can take advantage of some parallelization here. So if now you had k computer processors, we could test k models at a time, and then we could complete our grid search in n over k steps. Another form of parallelization is called bulletin board parallelism, and this would allow you to do an iterative search with a population of models, and evolutionary algorithms can be implemented with this kind of style of parallelism. And so here, if you had k processors, one of your processors acts as a controller, and it posts jobs to a bulletin board. And then the workers can take jobs off the bulletin board, run simulations, and then report back to the controller. This was the result of this simulation. And so here you could have you post a bunch of jobs and then divide them up the workers. And so every j over k steps, now the controller could take all those results and see, where are we in this parameter space? How are we performing? Let me generate a new population of models to test next. And so after some number of generations, then we will have tested n models. And again, this would have been done in n over k steps. But like I mentioned, in order to even evaluate a model that has many features, there might be a lot of little tests that we need to run even to evaluate a single model. And so here's sort of an example of another one of my favorite types of experiments that's looking at synaptic integration in neurons. So here we can take a little stretch of neuronal dendrite, and we can actually activate individual synapses one at a time with 2-photon glutamate uncaging. And so we can sample what's the amplitude of the response to each of these individual synapses. And then we could activate them together, and we can ask, does the neuron do linear summation and just add up all of these unitary responses, or does it do something different, something nonlinear? And so we can compute an offline sum of all of these synaptic responses, and then we can stimulate multiple synapses together and look at the actual response. And so what this group found in these dentate granule cells is that the neuron sums its inputs slightly superlinearly, so more than the sum of the unitary strengths. And then they wanted to get at the mechanism. And so they repeated these experiments with a blocker of NMDA receptors. And you find that that was contributing to this nonlinearity, that if you block NMDA receptors, which are these voltage-gated glutamate channels, that now synaptic input summation becomes sublinear. But if you think about this, the experimentalists had to perform dozens of small little tests and experiments in order to arrive at this single curve. And now, like, the feature that we want our model to reproduce is maybe the shape of this curve. But in order to do that, we would have to run dozens of tiny little simulations. And so if we're in this scenario where we not only want to test n models, but in order to even evaluate one model, we need to run a bunch of small simulations. We really need a parallel computing approach that is hierarchical. And so I'm calling this nested parallel computing. And so here what we would do is we would have K processors, excuse me, and then for each of M generations, our controller would post J jobs that need to be done. And that would be that we want to evaluate P models, but for every one of those models, we need to evaluate, we need to run L simulations to compute our features. And so then each worker would now perform one test of one model at a time, and it would report the results to the controller. And then the controller can decide a new population of models to test. And so then at the end, after M generations, you've now evaluated your N models, and you've done it with N models times L simulations divided by the number of workers. And so this is an approach that could be used to try to tune a model to reproduce this kind of synaptic integration phenotype. So we had to run a bunch of simulations where we stimulated one synapse at a time. Then we ran them where we stimulated more than one synapse together. And then we repeated it all with an NMDA receptor blocker. And so this approach can be used to tune this kind of complex feature of a neuronal cell model that requires that many simulations be executed. And in order to do this, I needed an algorithm that would be a little bit more efficient than the standard evolutionary approach, because like we showed, it took 1 million model evaluations to get good performance out of an evolutionary algorithm. And if we add on top of that, there's 100 simulations that need to be run for every model. Now you're talking about 100 million or 1 billion simulations that need to be run. So I needed something that was a little bit more efficient. And so what I've done here is basically taken basin hopping or simulated annealing, which was really a single objective algorithm that was serial. And I made it into a population-based parallel algorithm that uses a multi-objective performance criterion to evaluate models. So I've really kind of parallelized basin hopping and borrowed some ideas from evolutionary algorithms. And so how this works is that for every generation, you evaluate a population of models, and then you rank their performance relative to each other on multiple objectives using a non-dominated sort. And then you generate a new population of P models, and you do that by performing just basin hopping style steps from all of the best performers that were in the previous generation. And then you reduce the temperature or the step size over iterations. And so it turns out that this approach actually converges in fewer generations than typical evolutionary approaches. And so that allows us to get good answers with fewer overall model evaluations. So here I'm showing you the total objective error for a single cell optimization that was really like a kitchen sink optimization. We ran a bunch of little tests. We were measuring features like firing rate, FI curve slope, spike rate adaptation, dendritic input resistance, synaptic input summation. We just took all the features that we care about and we tried to co-tune them all together. And so what you can see is that early on, we're generating models that span a wide range of objective error. And then over iterations, we're improving our performance on as many objectives as we can. And we're identifying models that are performing better and better relative to the other members of the population. And so this works pretty well and only required about 30,000 models be evaluated. And so during this optimization, what were we doing? We were varying parameters. So here, for example, was the dendritic sodium channel conductance density. And in the first generation, we sampled a wide range of the parameter space, and we found models that that were not performing very well. And then through the course of optimization, we sort of narrowed our search and more finely searched around this space near the best performing models. But you can see that this algorithm sort of follows multiple paths. It's always trying to identify, is there any better solution that's nearby any of these previously identified kind of local minimum? And then this parameter influenced, for example, this feature, which was attenuation of spikes from soma to dendrite. And then here we had the NMDA receptor conductance at synapses, and we varied this parameter until we were able to match our experimental target for the gain or the slope of this nonlinear synaptic integration feature. Okay, so now I'm going to talk about an application of the same approach to a big network of these biophysical neurons. So again, the Allen modeling group had built this model of layer 4 of visual cortex. And the single cell models had previously been tuned up, and they're biophysical and morphological and based on experimental data collected. And there are multiple cell types. There were a couple classes of excitatory neurons and a couple classes of inhibitory neurons. And then the model was wired up according to experimental details regarding their connectivity. But then there were still some free parameters because each of these cell types connects with one another. And we don't exactly know the sort of functional coupling strength of these different projections. And so if your parameters then become how strong is the mean synaptic weight between these different cell types, and then your experimental targets that you want to match are the response of this network to a visual stimulus. So here, for example, we have all of these neuronal cell types. They're just sort of spontaneously active without a visual stimulus present. And then we provide an oriented moving bar that moves across the retinotopic space of this little patch of cortex. And then the neurons are responding to that stimulus. And you can see that because a lot of these neurons are tuned to the orientation of the grading, some of the cells are very highly active. They are selective for that orientation. And then other neurons are quiescent, and they would maybe prefer the orthogonal orientation of the bar, for example. And so the method that the Allen group used to tune this model is essentially the domain expert, trial and error kind of approach, where we use as much information as we can from the experimental literature to kind of find our parameter bounds, and we just kind of search a bit in those parameters, in that parameter space, to try to find a model that recapitulates the experimental data. But there's another thing that we need now, another trick. If we're running a big network model, we already need parallelization even to compute the response of the network. So if we had, for example, 1,000 computer processors, but we wanted to simulate 10,000 neurons, then we could gain some efficiency by putting 10 neurons on each of the 1,000 processors. And then all of those processors are collectively collaborating to compute the network, and they're sharing information about which neurons spiked at what time to update their state variables. So that's called collective parallelism. And in that scenario, you need Q processors to simulate one network model. So now we need Nested to do another trick, which is that if you have a bunch of processors and you're going to be doing this iterative approach of generating models, simulating multiple different tests of the model, now each of the workers actually needs to use Q processors to simulate one test of one network model. So this concept of nested is kind of hierarchical organization of parallel computing, so that you can have your iterating over populations, you're iterating over models, you're iterating over tests, and you're allowed to have these kind of little subworlds of collective parallelism that's doing the computation. And so, like I mentioned, the parameters that we're varying for this network model are basically the coupling strengths or connection strengths between the different cell classes in the network. And what we want to measure as features are the firing rates of each of these cells in different scenarios. So a spontaneous firing rate, the firing rate of cells when they receive their preferred stimulus or their orthogonal stimulus, for example. And so you can see that if we sort of sum up over all of our objectives here, again, early on in the optimization, we were sampling a wide range of parameter space that was leading to a range of objective errors, but we were able to improve our performance over generations and identify models that are performing better than their neighbors. And so the way that I parameterized this network was I basically considered the tuned model, the published model, to all have a value of 1 for their synaptic coupling strengths. And then my parameters were multipliers on those tuned synaptic weights. And so you can see that what this optimization approach identified, in many cases, these orange dots are very close to the blue dots, which is reassuring. That means that the optimization identified a model configuration that was very close to the sort of domain expert answer about this parameter. But in some cases, the optimization required large differences in the synaptic weights. And in fact, many of these that required some divergence were inhibitory synaptic strengths. And this maybe would have been a parameter that is a bit under constrained. It's like, what do we know enough about the functional strength of inhibition onto PV positive inhibitory cells? And then we have all of the features that we cared about. So here's all of the experimental targets, these stars, and then the performance of the optimized model relative to those targets versus the original model. And so in many cases, this optimized model performs similarly to the original one, and in some cases there's some substantial improvement, and in other objectives there is a bit of a cost. And so again, the end result of this kind of multi-objective optimization is usually that you get a whole family of models that have kind of different performance relative to different objectives. But this is really encouraging, because it means that if you sort of start off and you have a very complex model and you don't have a lot of domain expertise, you don't know where to start, that this is an algorithm that could, proceed quite efficiently, and in a small number of model evaluations could give you sort of a good guess and another place to start. And then you can always continue to use domain expertise and some trial and error tuning to kind of polish up the model beyond that point. So I think this provides a great sort of tool that we kind of needed to solve this problem of how do we tune, complex neuronal circuit models. And we need new tools and new algorithms to do this. And so Nested is a new Python-based parallel computing tool that I've developed that is for population-based optimization of parametric models. And population annealing is a new algorithm that is kind of a hybrid of basin hopping and of non-dominated sorting, and it enables efficient multi-objective optimization of compute-hungry models. So thank you for listening. This work was done mostly at Stanford with a great group of collaborators. Michael Hines, one of the developers of the Neuron software, really helped me out a lot in this project. And it was a lot of fun to interact with Anton and Kyle over the layer 4 model. And I have to also just mention that the fact that this model was implemented, with a nice API through the brain modeling toolkit made it very straightforward for me to sort of take a model that I was not involved in developing, but then sort of fitting it into the API of this nested infrastructure and being able to successfully tune their model. So you can find the repository for Nested on my GitHub. It's in a bit of a state of development. It's working, but it's not well documented. And so if you have a use case for this kind of approach, please reach out to me, and I would be happy to work with you to see if this can help you in your research.